for having me. Today I'll be sharing with you a bit of our work on a new technique to engineering very efficiently patient-derived mutations in either KAT6A or KAT6B and even some other genes, which is helping us to understand how different mutations in KAT6A and B are leading to pathogenesis. So a bit about myself. Have a background in biotechnology engineering, and I am currently pursuing my PhD at Dr. Xiang-Jiao Yang's lab in McGill University. He's a leading expert on epigenetic regulators, and his lab has made major contributions into the understanding of these epigenetic regulators' work in or role in disease and development. Recently, our current research goal has been to bridge molecular biology techniques with the understanding of rare genetic diseases, which is what brings me here today. So before diving into more experimental side, I wanted to go quickly over some key concepts, starting with what is a gene mutation. And a gene mutation is simply a change in the DNA sequence that can disrupt how a gene functions. Sometimes it can have no effect, or sometimes, like in the case of KAT6A and B syndromes, this can lead to the whole starting of a pathogenesis. And we can further divide mutations based on their origin. We have inherited mutations, which means that they are present in one or both parents and they are passed down to the kid. And this also means that all the cells carry these mutations since the moment of conception. And then we have what we call de novo mutations, meaning that they are new, they are spontaneous mutations that are not present in either of the parents. And this is important because this means that these mutations happen in the early phases of development. And this also implies that the majority of them have a very low recurrence rate. However, this is important because in the majority of KAT6 syndromes, the mutations that we see are de novo, and this arises a new challenge for early and accurate diagnosis. So to just refresh a little bit over what's the role of KAT6 genes, where they are lysine acetyltransferases, meaning that they are enzymes, which also means that they are proteins able to catalyze a chemical reaction. In this case, they are tiny chemical groups known as acetyl groups to histone proteins. And we can think of histones as spools around which DNA is wrapped. And once you add these tiny chemical tags, they become more open, and this allows for genes to be activated. So any mutations in either KAT6A or KAT6B can lead to a disruption of this regulation process. And this is especially important for neurodevelopment. However, KAT6A and B, they don't work alone. They are part of larger protein complexes, as we have already been going through. These complexes are known as histone acetyltransferases complexes, or MOS and MORPH. And where different protein partners like BRPF1, ING4, RNG5 support them. And then we also have EAF6, which serves for structural support and also helps in the transcriptional regulation. And it's actually being part of this complex what allows KAT6A and B to do their acetylation reaction in histones. So any disruption in KAT6A and B can lead to the can destabilize how this complex work, or even if it's for a form at all. And this can happen through multiple mechanisms. It can be causing the protein to not be able to form correctly, which would impair its enzymatic activity, or maybe it's affecting one of the binding sites for the protein partners, or maybe it is affecting the way it can bind to DNA. To further understand how can a single mutation can cause a whole disruption into the complex formation or its function, we can look at them at a more molecular level, these mutations. We have what we've been talking about all morning, missense mutations, meaning where a single DNA base pair changes or a letter in the DNA, and this can sometimes lead to a non-phenotype scene, or maybe it can lead to a complete disruption into the protein function. We have then more what tend to be more severe mutations, which are nonsense mutations that can lead to either truncation forms or frame shift changes. For the truncation, this means that there is an early subsignal that produces an incomplete form of the protein that in the majority of cases can be non-functional. Or we can have even more different type of mutations like deletions or insertions that basically throw off the whole reading frame of the protein sequences. And all these mutations and even more complex ones can happen in either KAT6A and KAT6B. What also influences the severity and the characteristics of the syndromes that we see in either KAT6A or KAT6B is the location of these mutations. And as you can see here, mutations can happen in all of the protein domains. And the difference between them are also correlated with the phenotype that had been identified. In here, there are some of the mutations that have already been identified and correlated with specific phenotypes of either KAT6A and B. However, these are not all the mutations that exist. In fact, as more patients have undergone genetic testing, we have been discovering more and more derived mutations in KAT6A and KAT6B, and many of them are classified as variants of uncertain significance. And this means that we don't know yet, or we don't have scientific or clinical data to support that these variants are actually causing pathogenesis or the clinical relevance of them. This is a screenshot from a CleanBar database, which is a variant public database. And if you type in KAT6A, you can see that almost 600 of them are classified as variants of uncertain significance. And this is important because our role as researchers is to figure out which of these mutations can actually lead to pathogenesis, and more importantly, why. Because this limits our understanding of the disease and the underlying cellular and molecular mechanisms that are causing the phenotypes that we see. And this also limits clinicians and families that need to interpret the specific variant to give a proper diagnosis and a proper course of treatment. So this is where a powerful molecular biology technique comes in, that it's called site-directed mutagenesis in vitro, that basically allow us to recreate patient-derived mutations in the lab. So what is exactly mutagenesis? Well, it's basically basic molecular biology lab a technique that allows us to intentionally introduce a DNA change in a sequence in a very precise way. So what we do is that we look for patient-derived mutations, and then we use mutagenesis to reproduce them in the lab, and then we test them. We usually do this by acetylation assays. And then we can actually see if this variant specifically is causing a disruption in the gene expression, or it's causing a malfunction in the protein function, or maybe it is affecting the way the complex is formatted. So this is a very powerful way to ask if this variant specifically is causing or leading to disease. We can think of mutagenesis as the word in a word file, the tool for research for find and replace. But in this case, instead of uh, changing a word, we're basically editing DNA. So this is a bit diving more into the more experimental side, but I think it's important to understand how mutagenesis works. We basically have a vector. A vector is a carrier for a DNA sequence. In this case, it's for either KAT6A or KAT6B genes. And then once we have these vectors, we can design primers. Primers are basic, very short DNA sequences that act as patches of DNA, and they are very useful to start a PCR reaction. Actually, they are needed for it. Then we can run a PCR reaction, and because these primers carry our mutation of interest, when the DNA replication process is happening, the new vector will now carry our mutation of interest. Then we move on into digestion step, in which will allow us to separate the templates that were used, the vectors that were used as templates, meaning that they don't carry the mutation of interest, and now the newly mutated plasmids that were engineered thanks to the primers. Then we move on into the transformation into bacteria. This allows us to recreate millions of copies of our plasmid of interest that carries the mutation. And then we do a process of extracting this DNA and screening for colonies to see if actually our mutants carry the mutation that we engineered. And this methodology is based on a quick change method that it's like the gold standard for mutagenesis, that it's been mainly used in all the labs around the world. And this specific method has two specific characteristics. First is the use of very long primers that carry the mutation right in the middle. You can think of these primers as being like mirror images of each other. And this specific design have some challenges, especially if you're using very large DNA templates, like is the case of KAT6A and KAT6B genes. And it also uses the PFU DNA polymerase. That when it was this method developed in the 90s, it was one of the best DNA polymerases available in the market. However, we have now newer and more effective templates for this. So basically, the challenge with this method is that it's very slow and unreliable for the way we need it to work. It can take from weeks to months to recreate a single patient-derived mutation in the lab by using this method, which is clearly not time or cost efficient. So this is where our current work comes in. We have been systematically optimizing the PCR conditions, the type of polymerases, as well as the primer design of the quick change method. And this has led us to the creation of the P3 and P3A methods, where the P3A is basically an improved version of the P3 method. And this name makes reference to our primer design, which we consider is the smarter design based on protruding 3' overhangs or arms. So this design, what it's basically is we still have a complementary region that carries the mutation in the middle, but then we have a flanking overhangs or arms that make the DNA synthesis much more reliable or less prone to errors during the PCR reactions. And then for the P3 method, we tested derivatives of the PFU polymerase, meaning PFU ultra and PFU fly. And by using them, we were able to increase the efficiency up to 50% average. So this still leaves some room for improvement. So for the P3A method, we tested the Cube5 and the Super5 polymerases, which are the best two commercially available polymerases at the moment. And by using them, we were able to recreate variants with up to 100% efficiency. So both of these methods proved to be very fast, cost-effective, and highly efficient for engineering KAT6A and B mutations. And what it's even more important to mention here is that this high efficiency seen with the P3A method led us to now be able to use this for what we call cassette mutagenesis, meaning that it's a new way to now edit DNA in vitro. And in this case is to delete up to 5 kilobases of DNA fragments, or also place insertions up to 0.5 kilobases, or even do complete fragment replacement without the need to rely on recombination or CRISPR techniques, which opens a whole new landscape for fast and cost-efficient DNA editing in vitro. For the specific case of KAT6A, we were able to engineer multiple patient-derived mutations. In the case of KAT6, we recreated 10 different mutants in the lab with an average efficiency of 80%. And for the case of KAT6B, we were able to recreate 9 different mutations with up to 96% efficiency. This also proved that both methods are reliable for its use with large DNA plasmids. Both of these vectors are almost 12,000 base pairs. And even more importantly, that we were able to engineer all of these patient-derived mutations in less than a week time. In comparison, by using the quick change method, we were not even able to obtain any bacterial colonies to screen them, even after multiple trials in the lab. And even though I've been talking about the use of these methods for KAT6A, its applications go far beyond that. We successfully use the P3 and P3A mutagenesis methods to engineer mutations in over 20 different expression vectors, covering more than 100 different mutations. This includes not only epigenetic regulators like KAT8 or P300, but even viral genes. We tested these for vectors encoding for the spike protein from SARS-CoV-2. And because of this versatility, the method can support research in a wide range of fields, from cancer biology to other neurodevelopmental disorders and even virology research. It's also a great tool for synthetic biology and personalized medicine, where precise DNA changes are required. Especially the P3A Cas6 mutagenesis methods now allow us to study further molecular characterization of protein domains, also on tag or tag proteins for different studies, and even use them for the insertions of specific plasmid elements such as LOXP sites or even guide RNA cassettes for CRISPR systems. So to wrap up, I would like to leave you with three key points. First, our P3 and P3A mutagenesis methods are designed to be fast, reliable, and versatile, which solves many of the issues and challenges seen with engineering multiple patient-derived mutations in large DNA templates like KAT6A and B genes. Second, these tools empower researchers to study patient-derived mutations more efficiently, which accelerates functional testing and variant interpretation. And finally, this work helps bridge the gap between basic research and clinical diagnosis, which moves us closer to target therapies for rare genetic diseases. Before I end, I would like to take a moment to acknowledge all the people and organizations that were behind this work. First, thank you to the KAT6 Foundation for offering us the opportunity to share our work at this conference. I'm very grateful for being part of Dr. Xiang-Jiao Yang's lab, which he usually comes to this conference, but this time he was not capable to. And also to my lab members for also their work for all this presentation. And of course, thank you for listening. I'm glad to answer any questions. Thank you so much. Yeah, those where we usually work with vectors that we have been using since a long time in the lab. The majority of them are either derived from the gene plasmid repertoires, or some of them we use from collaborations with other labs. Yes. Y- yes. Well, we only actually use the bacteria to make a lot of copies of our plasmid of interest. Once we reach a specific concentration, we extract this. So now we have a mutated version of our plasmid carrying a different variant of KAT6A and B. And then we do what we call functional testing. We use as human cell lines to express a specific variant in the lab, and then we run acetylation assays to prove if these new variants are causing acetylation defect on which we detect with antibodies specifically for histone marks. Okay. We use Yeah, we use mammalian expression vectors because we use HEC293 cells. But what we have been found is actually these methods should be applicable to any kind of vector. Yes. Okay. Yes. Yeah, and also we now working also KAT6A and B. They don't have such a high GC content, but we have other plasmids such as the KAT2B that have very high GC regions. So this made them incompatible with the P3A method. We'll be publishing it soon on how to improve um, mutagenesis efficiency for templates that have very high GC contents. If this is the case for the gateway vectors. Yeah, and sometimes it's because of this. And yes, and sometimes it's not even that it's the variant that it's actually causing having these very high GC sequences. In some of the cases, it's actually the plasmid elements. We have seen this with ARIS elements or even with some uh, promoters like the what's the name of this promoter? Well, promoters are very widely used for mammalian expression systems. Yes. Thank you so much.